So of course, what kind of neuronal structures are in white matter again? Not myelinated axons. And basically what's in the gray matter around it then? Basically everything else, yeah. Cell bodies, dendrites, unmyelinated axons, axon terminals, etc. So the key thing with the brain anatomy is it's inverse to the spinal cord. The gray matter's on the outside, the white matter's on the inside. But this is the key thing. What you're going to learn today in lecture, and this is where the statements come into play, but I love talking about 15 because it's all about just dividing the brain up into sections, and basically each of the gray matter sections of the brain have a specific function. Now when it comes to general purposes, frontal lobe, motor functions, posterior area for sensory functions, and when we start talking about the visual area or the auditory area, these are areas dedicated to processing that kind of information. But the key thing you have to realize is that when it comes to anything, and I always like to use food as an example, but when you look at your just favorite dish that your maybe mom or dad or whoever prepared for you guys, obviously you obviously have visual information coming into your brain, you have olfactory information coming into your brain. When you taste it, you have gustatory information coming into your brain, and even when it hits your maybe tongue, you have tactile information coming into your brain. And of course, when you think about food with all those senses coming in, can a common sort of overall thought to that food come into mind? Again, one of my favorite movies to exemplify this was Ratatouille. You guys remember Ratatouille? Mm-hmm. Remember when Egon was eating the dish that the mouse prepared at the end, and he was transported back to his childhood because it invoked all those memories? Well, that's amazing what the brain can do. Basically, they will take all this information, even though this information is processed in different regions of the brain, all the white fiber tracks basically connect all these areas of the brain together, and of course, you can come up with some common theme or thought about anything, all right? And it doesn't have to be food. It could be when you look at a person or hear music or whatnot. Basically, your brain is communicating with other parts of the brain to invoke some sort of overall common thought or image or whatever about that object, person, whatever. But in this case, you have to have that communication, so all the white fiber tracks in the brain will communicate with all the gray matter regions. And that's why when it comes to some emphasis today, we'll talk about association fibers, we'll talk about comissural fibers, we'll talk about production fibers, but those are the three types of white fiber tracks that you find within the brain to allow all this communication to occur. Okay? And again, you can see some of them. So in this case, let me get a knife and let's do a fresh sagittal section through the brain. Because this would be a lot better than this. No, actually the dried up one is not bad, actually, I have to admit. So I can actually use either one, but a brand new one is probably going to be a little bit better in this case. Take out the fresh brain. And all we're going to do is do a straight sagittal cut down the midline. And in this case, like I said, the one thing lacking from this brain is the dura mater. So, no dura mater to deal with, so no falx cerebri, no tentorium cerebelli, whatnot. So basically that's what the brain looks like, and again, I want to make sure that it looks like what you see in the diagram up there. So we go through all the numbers, so I pinned them all down. Basically, number one is in the cerebrum or cerebral cortex. It does refer to the gray matter, and again, that gray matter is composed of that motor region, sensory region, and today in lecture we'll divide those up into the specific regions, whether we're talking voluntary movement or visual processing or auditory processing, whatnot. And so pin number one can go on either side, but I will match it, so we'll go into this side right here for the cerebral cortex or cerebrum. Number two is the cerebellum. It's this big cauliflower region back here. And again, the nice thing about the cerebellum is you can also see the extensive white fiber tracks within it. It does tell you that when it comes to this integrative function of the brain, that all different parts of the brain must communicate with one another to keep you alive. And one of the key things about the cerebellum is for things like posture and balance. So even though you think about skeletal muscle, again, you guys learned skeletal muscle is voluntary, but remember, not 100%. Your muscles are being controlled by your brain to make sure you maintain proper posture and balance. Again, right now, if you think about it as an adult, do you ever think about falling over, or do you just walk straight and stay upright? Right? You just walk straight. Same thing when it comes to my favorite skeletal muscle is the diaphragm. You ever have to think about breathing, or you just breathe? Yeah. And again, it's controlled by autonomic centers in the brain. So number two, also on this side, we'll put pin number two in the cerebellum. All right, number three, corpus callosum. In this case, this one's a little bit harder to see, and I don't have a pin number three.
Okay, I'm switching in. Closer. Someone figure out how to post this on Blackboard.
Let me go get my label maker. I have a 3.
Because I don't know how to do that.
Just post it on YouTube and then post on Facebook.
Yeah. You do it.
Send it to your email. He's making a number 3.
You can just write number 3.
You got a big boy.
Close enough.
This is the most wasteful label maker in existence, by the way. Look at how much it wastes. Hey. Yeah, that's perfect. Alright, so 3. Any other number I'm missing? Do I have a 5? I do not have a 5. Do I have a 12? I have a 12. Do I have a 14? I have a 14. Do I have a 17? 17. 23 I have, and a 46. Good. All I need is a 3. Thank you, Amanda. Big 3. Alright, so number 3 is supposed to be in the corpus callosum, and that is one structure, hint, hint, that you guys should know for any assessment and or ID. Again, I mentioned the cerebrospinal fluid yesterday, and in your notes and in the lecture yesterday, I mentioned that when it comes to cerebrospinal fluid, it's formed in your brain ventricles. Well, your brain ventricles are chambers in the brain, and in this case, it's easy to find the corpus callosum because the corpus callosum actually forms the roof of your lateral ventricles. So on this brain right here, actually, let's do this first. Okay, so if I take all the pins out of this brain, so we can do some visualization before I pin it on the other one. Again, there's your brain, but if I spread this out, can you see those big chambers that I'm spreading open right there?
Mm-hmm.
So everybody see those chambers? Okay, and again, it's the same on this side, but basically those are the lateral ventricles. And basically that's where your cerebrospinal fluid is formed. Ependymal cells line those chambers, and of course those are responsible for converting blood plasma into cerebrospinal fluid. But now, if you look at this portion right here, again, just that white fiber tract right above the lateral ventricle, that's your corpus callosum. Okay, it's what we call a main comajural fiber because that structure allows communication between the right and the left sides of the brain. And so in this case, we can pin it on either side, but I have it on this side, so we'll pin it right here. Now, for those of you close enough, this is not a structure you have to identify on the brain because, first of all, sometimes it gets destroyed very easily. Plus, it is labeled on the actual skull over there. It's actually labeled on the skull over there. And in this case, if you guys can see it right here, when you come up, there's a thin membranous structure over this lateral ventricle, and this is called the septum pellucidum. So even though you have a paired set of lateral ventricles, the lateral ventricles are separated by a membranous structure. You don't have to worry about identifying it on the sheep brain, but you should be able to identify it on the model because it is labeled there. And in this case, again, just like your quiz today, when it comes to identifying these structures, depending upon the detail, so to speak, I will give you some functional lead-ins. So if I say, again, in this case, identify structure number 3, which forms the roof of the lateral ventricles, that's an anatomical clue. Or I could ask you, please identify structure number 3, which allows the communication between the right and left hemispheres of the brain. Then you should be able to identify it as the corpus callosum, because in this case it's relatively small. Same thing when it comes to things like the septum pellucidum. It's just a white area on that model with a number on it, and so if I want you to identify the septum pellucidum, I would say, please identify this membranous structure which separates the lateral ventricles. That's the key for you guys to identify it as the septum pellucidum. Okay? So number 5, lateral ventricles, just the whole. So where's my... Oh, thank you. Thank you, Heather. That's not the one that's coming out. It's the, there it is. It's the wasteful label maker. And so what I'll do is, since this side doesn't have the septum pellucidum, I'll just put pin number 5 on that side like it is on the picture. Thank you. Number 12, medulla oblongata, and you guys should already know the function of that area of the brain, right? It serves as what? At least
Rhythmicity for your breathing.
rhythmicity for your breathing. It also holds other functions like your cardiovascular control center, so it also helps to regulate things like blood pressure. So it goes right there in the brainstem. Number 14, the intermediate mass. In the case, it's called the intermediate mass in the sheep brain. Don't ask me why for the sheep, but this is equivalent to your thalamic region of your brain. So it's just this big round guy right here. And in your case, the key thing about the thalamus is that even though we bring in all the sensory information, we see things, we hear things, we taste things, all that, the thalamus is actually the portion of the brain that receives all that information first. So even though you have a visual cortex, when you see things, that information is not directed directly to that visual cortex. It gets shunted to the thalamus first, and then the thalamus sends it to the visual cortex. The way I always like to think of the thalamus is like the post office. All this information comes in, but it's the job of the post office to send the mail to your correct addresses. And so in this case, all the sensory information comes in, and in this case, it's parlayed to the right portions of the brain through all those fiber tracts we've talked about. Number 17 is the third ventricle. In this case, the third ventricle lies right above the intermediate mass, and I have it pinned on that side, so I guess I'll leave it here as well. And the neat thing about the ventricles, especially when it comes to, let's see if I actually destroyed them. You can still make out some of the small blood vessels within these chambers, but basically 17 third ventricles right above the intermediate mass. Okay, and again, it's just a site for cerebrospinal fluid formation. Number 23, cerebral aqueduct, basically conveys the cerebrospinal fluid from the third ventricle to the fourth ventricle. So it's basically above the medulla and the brainstem, between the cerebellum, and I've pinned it out on that side as well, so we'll leave it here. So we'll just pin it right there. So I have to actually lift up the cerebellum to actually put the pin there, but there's that basically channel or canal that conveys cerebrospinal fluid to the fourth ventricle. And then number 46 will be your fourth ventricle, and this one is basically just smack dab in the middle of the cerebellum. So if you peel back the cerebellum, can you guys see that little gap that I spread apart right there?
Mm-hmm.
That's how small it is. So when you guys come up and take a closer look at the brain, literally you just spread apart that cerebellum right there, and the pin will just go right up that little indentation, and that's how small, and it's on that, no, it's on that, I'm going to put it on the other side. That side's getting a little bit crowded, so we'll put it on the opposite side right here. So that's the only pin that will be on the opposite side. But again, I spread it out a little bit. That's the fourth ventricle. Okay? But that's basically all the rudimentary structures you can see from a gross anatomical perspective on the brain. Here's that coronal section I was talking about. And when you look at this coronal section right here, you can actually see the corpus callosum and the septum pellucidum quite clearly when you pin it back. Okay? But I'm not going to give this one to you guys. The specimen that will appear on your third practical will be this puppy right here, okay? As long as it stays moist, I'll use it. But again, if I slice up a brain, or if you look at this one, you should be able to transfer the pins from this brain into this brain, no problem. The only thing, like I said, lacking on this one, the septum pellucidum is destroyed. And in this case, because it's dried up, can you guys see the fourth ventricle is a little bit clearer now? Mm-hmm. It's sort of shriveled up, but you can actually see the gap in the cerebellum. So in fact, sometimes the shriveled up brain is a little bit better to visualize the things. But either or, I'll leave these two side by side to see whether or not you can actually point and make the appropriate association, okay? But to be totally nice to you guys for your quiz next Tuesday, bottom line, know this picture, or sorry, know that picture, because that's what's going to be on your quiz next week, okay? If you know that picture, you will do very well on next week's quiz, okay? Big hint, very well on next week's quiz. Quiz next week?
That's the only picture that we have for ID for next week?
For your quiz?
Is there
No, there's a PowerPoint presentation, so there's all the pictures of that, but for your quiz specifically next week, no that picture. No that picture. So, all right, so any questions on that? That basically finishes up all of these specimens you guys need for your third practical. Like I said, I got extra brains in there if you want to cut up your own, whatever. But obviously, get to know the one we've pinned out here right now, because this is the one, as long as it stays moist, we'll use this for your practical next Wednesday. Okay? Right. Any questions? Other than that, let me grade your quizzes. Obviously, just study, study, study. And you guys are good to go.
Wait, let me just finish all the way around, and then someone figure out how to post this, and then you guys can all take pictures.